Share. Yes. Okay. So, good afternoon, everybody. Welcome to this departmental seminar, and I'm very happy to have Analabha with us. Analabha did his masters from ISI Kolkata and also PhD uh, in biostatistics or genetics in ISI from ISI Kolkata. Subsequently, he went to Stanford and then University of California, and then he came back to India to join this National Institute of Biomedical Genomics in Kolkata. He, by training, he's a biostatistician, and we'll be talking more on the evolution of human being. So, Analabha, please.
Okay. Thank you, Rajiv. Thank you for this invitation. It's essentially a pleasure. It's a huge pleasure to talk to audiences who do not encounter possibly any of this in their daily work or in their daily research. So the plan of today's talk is very, very basic in a sense that I'll start with things which are very basic. I'll try and give examples which are very basic. But the whole idea is to try and narrate questions or try and narrate stories which have huge implication in terms of understanding who we are and implications in understanding how we can modify ourselves or how our lives, possibly our society as well. So I will start with this quote. This essentially is a title of a paper by Dozansky, which says that nothing in biology makes sense except seen in the light of evolution. And I'll be talking about evolution, mostly about human evolution. By human, again, I'll traverse a little bit about what I mean by human. But this is primarily about anatomically modern humans or Homo sapiens sapiens, the people whom we are here. So we are anatomically modern humans. So let's start with where I come from. This is essentially a cell. Generally, what we call is an eukaryotic cell. So the eukaryotic cell has a nucleus. Within the nucleus is this set of chromosomes which have DNA in them. Right? So DNA is called the molecule of life, and DNA is passed from one generation to another. So it contains all information which is important for inheritance, and because of this DNA, we are similar to our predecessors or ancestors. So the DNA is primarily within this, but there is also things like mitochondria, which is also called the powerhouse of cells, which also contains a little bit of DNA. So the mitochondria that we have, all of us, our mitochondria are inherited from our mothers. The DNA is 50%, roughly about 50% from our father and mother. So humans have 23 pairs of chromosomes. There are 22 which are autosomals, and one pair, which is called the sex chromosome. So if you have an X and a Y, then you are a male. If you have two X's, then you are a female. So this is primarily the pair of chromosome which determines an individual's sex. By sex, I of course I mean biological sex here. The chromosomes are like this, and this is a specific time in the cell. Those chromosomes are packed in different ways and can take different types of configurations. This is one of the configurations, and within the chromosomes, this is the double helix, the famous double helix structure of DNA. This is coiled within the chromosome in a very, very organized fashion, and that in itself is a fantastic problem. Maybe I'll talk a little bit about that problem a little bit later. So just to give you a sense of how things are, the genome has 46 chromosomes, 23 pairs, 23 into 2. It is approximately about 3 billion base pairs. The bases are the A, T, Gs, and Cs, which are the building blocks of this. and they're so if you take a cell which we cannot see through our naked eye, most cells we cannot see in our naked eye. If you take out the nucleus and if you take the DNA out of it and just stretch it in a straight line, then that's about 6 feet long. So the DNA is actually supercoiled within your nucleus in a very, very efficient way. Because if you think about this, then all the functions that we do is essentially some kind of information which is being coded, right? The functions are determined by proteins, and our genes actually determine which proteins to code under which kind of situation. And that have very, very different kinds of specificity, which is like the proteins that are coded in my brain is not the same set of protein which is coded by my immune system, or the same set of protein which is coded by my heart. So the DNA remains the same depending on which tissue it is, but it codes for depending on which depending on the function of the tissue. And not only that, the more important and interesting part of this is just imagine the scenario here, which is that we are here. I'm talking. So there are specific kinds of proteins which are being expressed in my cell. But just imagine that if something happens here, like an earthquake, then all of us would immediately run, right? Or some other kinds of programs will get initiated, which would mean that a completely different set of proteins will get initiated. So just imagine this, there are 3 billion bases which are packed within this nucleus, and at the same point of time, or in a very small instance, in a very small interval of time, you shift from a situation where you're listening to a lecture or giving a lecture to something which tells you to run, which means that this is an amazing information retrieval problem. This is superpowered, this is packed somewhere, but somehow, given the external stimuli, you can still go and get that particular protein in that particular moment, such that that translates into the kinds of programs, or that translates the kind of activities that you are going to do. And if it changes rapidly.
I have a question. So what would be the average time of expression of a protein?
What would be the average time of expression of a protein? It varies. It varies under different kinds of proteins.
A few seconds. Is it a few seconds?
Of course.
It's a few seconds.
Of course, it has to be. I mean, some of these has to be, right? I'm not running, but just imagine that I have to run. That means that there has to be proteins which gets into my muscles, which immediately needs to get activated, right? So that is that. So for that particular instance, those are almost instantaneous. That has to be instantaneous. So there are different kinds of proteins. There are certain kinds of proteins which are like continuously being expressed, right? Because I'm breathing, because there's a continuous amount of metabolization which is going on, etc., etc. But when there's a stimulus, when there is an external stimulus to enhance that immediately gets enhanced to a very can get enhanced to a very
some structure is highly densely boiled.
Yeah.
Retrieval of that protein sequence.
That's the reason I think that this is a fascinating problem.
But that will be done in seconds.
That will be done in milliseconds, essentially. So yeah, so because you have to first identify that, right? And that also has to be very, very specific, right? The function that starts in your brain is not necessarily the function that starts in your limbs, but there has to be a synchronization of what needs to be done, and that is done very, very fast, very, very. So these are the building blocks, the A, T, G, and C. So A, T, G, and C. These are like the alphabets of the DNA. All information is coded in sequence of these A, T, G, and C's. Depending on what kind of sequence or on a specific region, you code specific kinds of proteins. So if you look at this, then humans are essentially very similar to one another. So if you just take a random segment of 2 human genomes, then they are about 99.9% similar. Given any 2 individuals sampled from any 2 places in the world, roughly they're about 99.9% similar. Which means that if you have just one individual, then everybody is kind of 99.9% similar. So there is a reference genome of individuals, and every individual is very close to the reference genome. And if you imagine that your father and mother are 2 individuals, then they are 2 unrelated individuals. Both of them are pretty similar to the reference genome. So your genome is essentially like this, where just having the reference in most places is enough. There are certain places where your father or mother are different. One of them might be same as the reference, the other might be different from the reference, or in some of the rarer cases, both of them are possibly different from the reference. So in this genome, the genome which is 3 billion bases, there are only a small proportion of regions which is different from the reference. So as you can imagine, this entire data can actually be reduced to only these places which are colored red, where you have something other than the reference. So your genome can actually be summarized or reduces to a very small, much smaller proposition in terms of data, because you can get rid of what the reference is. So for an individual's genome, you can only look into places where the father and mother are either different from one another, or they're both different from the references. So having the reference has been a very, very useful thing. So our data essentially, our genomes essentially look like this. And our genome is our data. So as Rajiv said, that I'm trained as a statistician, and what I do is I use this genomic data, I use this kind of data, and I use this kind of data to infer about human evolution, human past, the paths people have taken in migration, and how populations have moved from one place to another, at what time points did they move, and what kind of interactions they had in the whole process. But I'm not going to talk too much about my work. What I'm going to talk about is integrated work of a lot of individuals, and of which I also have some part. Interesting part of genomics is that genomics is associated with diseases, but genomics actually gives us variation. This is one of the more apparent things which we know, that this is skin color. Skin color, of course, you all have a sense that skin colors have a very strong heritability, which means that we resemble our parents, we resemble our ancestors to a very large extent in terms of skin color. And if you look at skin color across the world, then there is a huge amount of variation. This is true for all kinds of things. So variation stands as a very important hallmark of all of this. Variation and diversity are things which has to be appreciated very, very early on in this. Genomics is oftentimes always generally associated with things which are very, very functional. But there are things which is like clasping of your hands. So if you just clasp your hand, you'll see that one of this, putting the right thumb on the left thumb versus putting the left thumb on the right thumb, one of these feels a little bit more comfortable. You can just do this. You can just ask your family members to do this, and you will see that this is something which has a heritage. So if you're more comfortable putting your left thumb on the right thumb, it is more likely that people who are related to you are also more comfortable putting the left thumb on the right. Has no possible consequence in terms of functionality, but these are things which are also genetic. There are things like rolling of the tongue. Some of us can roll our tongue, some of us cannot roll our tongue. This is very, very genetic. Again, possibly have nothing to do with functionality. It just gives diversity. It just gives variation in the entire population. So genomics is not necessarily associated with things which are like diseases, etc. It is also associated to all kinds of variations that we observe. And that can be innocuous things like clasping of thumbs or rolling of your tongue. So if we move into how people have used genomics, or how people have used this kind of genomic data in trying to infer about past, then the first thing that comes in our mind is the observation which was made by Margolis as old as 1963. So they were looking at cytochrome C, which is a part of what we call is what is more like a hemoglobin in us. So they were looking at cytochrome C. They were essentially looking at proteins in cytochrome C. And what they observed is that if there are inferences about speciation. So there are two species which are most closely related. There are species which are more distantly related, etc., etc. And what she observed is that if you just look at the cytochrome C and you count the number of differences, that can be correlated with the amount of separation time between species. So apparently there is a clock which is ticking in us in terms of evolutionary time, right? The clock is ticking. The number of mutations a species accumulate over a certain period of time is essentially somehow related to the time through which they have been separated. So just to give you a sense of this, this is me when I didn't have a beard. Of course, I think you can recognize this. I'm very close to a chimpanzee. So if you look at my DNA sequence, your DNA sequences as well will be 99.5% close to chimpanzee. So we are pretty close in terms of our relationships. But whoever tells you, we are pretty distant from a cow. So if you think about the separation times that we have here, this separation time is somewhere around 6 million years. However, this separation time is about 80 million years. So we are separated from a cow by at least 80 million years, which is again, I mean, not a very close time point in order to get the cows close to our so close to be our mothers, etc. So how does it reflect in your DNA? So the way it reflects in your DNA is something like this. If you just take random chunks from the same region, then this too will be either a human or a chimpanzee, and this will be a cow. And the cow will be very distant, very different from the human and the chimpanzee. So this is, suppose, my DNA, and this is a chimpanzee, whereas this should be the cow. So this is one of the basic premises which we use, which is essentially counting the number of differences that we observe. Of course, I'm simplifying things. This is not the only thing that we do. There are other kinds of things that we also do. But this is one of the major and possibly the most important intuitive idea that has gone behind all of this, which is that if you take two individuals who are related to one another, who are close to one another, they have fewer number of differences in the DNA. If they're more distant from one another, they will have much higher number of differences in the DNA. So you can relate this, you can take this a little bit more forward. These two individuals, they have same parents. So obviously they will have very similar kinds of DNA because they are inherited from their parents, but still there will be some differences because of different kinds of reasons. So their DNA will be similar, and it will be distant from somebody who's a Maasai Mara woman from Africa, right? So analogous, in an analogous sense, if you look at this, then this is the same way in which that these two individuals for certain regions of the genome will be very, very similar, and there will be fewer number of differences between them. And if you look at this individual and this, then this individual will be considered different.
So when you say a region, so how do you map, like, how do you say that this is the region I should?
So if you think about this, that this is 99.9% similar, then if you take a stretch of DNA, if it is suppose about 20 base pairs long in 3 billion bases. So if you just had 20 base pair long sequence, it kind of uniquely maps on each. So 20 base pair means it sounds. I mean, of course, mathematicians do understand it. So there are, each position can have just 4 observations, right? So it is like 4 raised to the power 20. So that's the number of the kinds of sequence that you get. But even if you take just a sequence which is of 20 base pair length, just random sequence of 20 base pair length, and if you map it across the human genome, it is very likely to map in only one.
So you're trying to maximize the intersection and then take the difference.
You maximize the intersection means it is almost like these are like homologous regions of the genome where almost everybody in the world will be very, very similar. Most people will be similar. Some people will have some differences. And this goes back to the whatever, if you construct the tree of life. So if you have genes which have very similar functions all over, so how do we understand that evolution is really happening from a genomic perspective? So if you look at the tree, look at suppose an amoeba, and there are certain genes in the amoeba which performs the same function that we do, very similar functions related to metabolism. Then the sequences of that particular gene in the amoeba is also pretty close to the same gene which we do. So this is true of the evolutionary time point of whatever 400 billion years. Sorry, 4 billion years, but or say 3 billion years. Even through 3 billion years, those regions have generally been used.
Use that as a common point.
We use that as common point. So we kind of align everything across. We align everything across the reference set.
You can, of course, acquiring mutations that come, but can mutations invert that? Get closer.
So yeah, I mean, so if you consider mutation something which is like a random event, and most mutations which are random event do not have anything to do with the select. That means that it just enhances diversity possibly, or have nothing which is functional. Then the mutation will just be there. And the probability of a random event in 3 billion bases to happen on the simultaneous place to go back is very, very low. So you can essentially ignore. So mathematically, you can consider this as something which has infinite number of sites. So the probability of a mutation happening in the same place multiple times is essentially zero. Though it happens. But yeah, it's very, very
Small,
very small. It happens. But it do not impact on the inferences that you draw. All right, so I'll start with the first story, the first debate that possibly this resolved. So this was interesting in a sense that when the molecular clock was first hypothesized, people tried to calibrate that against paleontological data, archaeological data that people have. The molecular clock, but I mean, as you imagine, that is dependent on discovery of a new fossil, discovery of anything which is painstaking. However, if you just imagine doing something like this, what you need to do is you just need to get individuals from right here, or species which are roaming around, right? You just get them, you just bleed them, get their DNA sequences out, and you reconstruct what the relationship is. So it's a much robust way in terms of data generation, in terms of validity, in terms of reliability, in terms of doing the experiment all over again, repetition of experiment, etc. So what now have become a gold standard is molecular methods. So the molecular methods are, so if you now find an archaeological thing which you are trying to substantiate archaeologically, then you try and validate that against the gold standard, which is DNA sequence. So one of the first questions that was resolved using this kind of technology is about human evolution, anatomically modern human evolution, so to say. So there were hypotheses about a multi-regional evolution of humans, which says that so there was one question which people said that it all started in Africa. I mean, Darwin was a big proponent of this, that it all started in Africa, and anatomically modern humans have started moving to or colonize the entire world starting from Africa. However, there are remains of people whose skeletal remains were like humans, right? You must have heard about Neanderthals, you must have heard about, suppose, Peking man, Java man, etc., etc. So these are skeletal remains, not necessarily anatomically modern humans, but these are from different parts of the world. And the multi-regional hypothesis would say that there are human-like populations all across the world, and the human population or the human variation that we see today are essentially locus by locus, multi-regional evolution of this. Versus the group of individuals who would say, no, it's all started somewhere in one place, and possibly that place is Africa from which it started. So one of the first things that looked into this is this paper, which is looking into mitochondrial DNA. As I said, the mitochondria is the one which is passed from the mother to the offsprings. So essentially, if you trace back the mitochondria, then whoever it is collected from, it goes back to your mother, to your grandmother, to your great grandmother, etc., etc. So it kind of goes back to the female lineage through which things have moved along. So what this people, this is a lab from Berkeley, what they did was they collected individuals from all across the world, and they did something which is like a tree, right? So the kind of things that we were trying to show here is essentially like a tree building. So there is a root, and then there are branches coming out of the root. So they essentially built a tree. And what they found is that this is the root. The ancestor is somewhere here. This is essentially a tree which goes like this. So you have to imagine this essentially like a line here. And as you can see, that the ancestor is here, and then there is one branch which separates very, very early. And there is another branch which comes here, and it goes all along. Now, if you can see through the, if you can see in this, see the levels of here, then the blue circles are essentially people who are from Africa. So as you can see, that all these individuals who come in this branch are people from Africa. And all these individuals, they come from all other places in the world, including some from Africa as well, but they come from Asia, Australia, New Guinea, Europe, etc. So Asia, Australia, New Guinea, Europe, right? So this is all across the globe. And if you construct the three of them, then they eventually coalesce somewhere here. And as you know, in this tree, the y-axis is essentially some proxy of time, right? As we are trying to make the point here, that these essentially means that this is a common ancestor to all of them, but this goes farther back in time. This is more closely related. So their common ancestor is more close. So this y-axis on the tree is essentially a proxy of time. So if you use the y-axis as the proxy of time, then you see that the Africans actually start their branching process way earlier than everybody else in the world starts. So this is one of the first papers which put forward very, very strongly in 1987 that the out of Africa is possibly the way you have to look into anatomically modern humans, because this is Africans. The way you start, you argue this is that these are Africans who are in some place, and these are Africans oftentimes are from, if you look at them, they have very deep branch lengths. These Africans can be like two people from two very adjacent villages, but they really go very, very deep in time in terms of their ancestors. And everybody else in the world from all other continents put together have an ancestor here, which is much shallower if you look at the y-axis, which is a proxy of time.
How are you? So I understand that you're making this branching distribution from the distances of DNA sequences.
Yes.
Kind of like that. Yes. But in how does that correspond to real time?
So that's essentially what the molecular clock essentially is. So if you look at them, that suppose if there are X number of differences, then the separation is about, say, 2 million years. So that was how it started. Yes. So if you go 80 million years, then that will be 40x. So this is.
But all these individuals that they picked up for the study, they are from a very on horizontal time slice, right? These are not old individuals.
Yes, but this is talking about ancestry. So if you look at their ancestor, they have a common ancestor somewhere.
No, I understand that aspect, but using this length of this y-axis, or in this case the x-axis, they correspond to real time.
Right. So if these two individuals have an ancestor, so if you consider these two individuals, their ancestor go back here.
But in terms of distances, I can understand this joining process.
Which means that their DNA, in spite of the fact that they are very close in geographical proximity, their DNA have a large number of differences.
So okay. Then my question would be: you have a graph which states how much distance will you incur in how much real time.
Yes.
So this graph has been generated by what means?
So this graph is so this is a so it started. It is generated through multiple ways. One method is that suppose you have a radiocarbon way of dating, okay, or other kinds of ways in which you date. So you look at fossil records. You know that this is the time possibly when the cow and the humans have separated, right? You have dated that to be 80 million.
But can you extract that DNA from those kinds of fossils?
No, you don't need to. You don't need to, because they had a common ancestor. So they had a sequence. The sequence has mutated across one branch. It has mutated along another branch. So the number of mutation is essentially your molecular clock, the number of differences that you observe at this time. So that's the beauty of using molecular data. That you can start with just extant individuals who are living now, and you can actually reconstruct that.
The point assumption that the mutation rate has remained the same throughout time and geography. Is it a valid assumption?
It is. Well, I mean, there has been questions about that, but it has. I mean, the rate of random occurring mutation, of course, has remained more or less uniform across. I mean, there is no observation which very, very strongly counters. Let me put it in this way. Obviously, we cannot go back and do this experiment all over again and do that. But there is no like anything else, like physics. I mean, I can say that what's the guarantee that the laws of physics were same 500 years before or 5,000 years ago, right? I mean, there's no way in which you can move backwards and validate that. But of course, there are ways and means by which the way we typically would like to validate that is that is there any observation which seriously questions this assumption? There isn't. All right, so this is the first paper which came out here. Then came a series of papers essentially from the same lab. So this is from a hypervariable segment of mitochondrial region. So they thought that this is not a functional region, so it should not be constrained. So it will accumulate random mutations. So that's the reason it is called a hypervariable sequence. And in 1991, they did the same thing. The tree, the algorithm to make the tree is slightly different from the previous paper. But what you see is again a much clearer distinction. In this particular figure, what you observe is that this open circles, these are Western pygmies from Africa. These are Eastern pygmies from Africa. These are the Khoisan people. So this is essentially this is pronounced as Klung. So it has this click sound in them, the click speaking people, which is essentially one of the earliest form of languages that people had. So as you can see again, I mean the Africans clustered together. This is again one of these trees which is circular in nature, but you have to imagine this, the ancestor is here. You have to hang the tree by this. And the Africans are together, and the non-Africans are here, and the non-Africans eventually coalesce somewhere here, whereas that is sister to Africans here, and it goes farther back in time. So they did two things. They also calibrated this. Why? And the calibration runs like this: that if you think about human evolution, they're according to their calculations in that point of time, which has been revised nowadays. So what they found is that it started somewhere in Africa in around 200,000 years ago. It's only 200,000 years ago that anatomically modern humans, the first evidence of anatomically modern human ancestor, the mitochondrial Eve, as they call this, was in Africa. And it is only about 60 to 80,000 years ago that people have moved outside. So more than half of the time that we are on this world, we as a species, we as anatomically modern humans, more than half of the time we have been only in Africa. There might have been unsuccessful movements outside of Africa, but that really doesn't count. That really doesn't have any evidence up until today that So the way this has possibly, the way that this possibly fits with the theory that fits with this kind of data is that we have been in Africa for a very long period of time, and then a very small group of individuals moved outside. They were pretty successful. They expanded in space and time, and then eventually colonized the entire world. This is 1991. In 2000, they came up with the complete mitochondrial DNA sequence, and they did the same thing. So if you look at this horizontal line, every individual on top of this horizontal line are non-Africans. All of these individuals are Africans. They also use an outgroup, which is our closest cousin, who are chimpanzees. And you can see that the Africans are here, the non-Africans here. All non-Africans, they coalesce somewhere here. So this star essentially is the ancestor of all non-Africans. And if you look at the y-axis again, the Africans go farther deep in time. And so these are like very closely related populations, which are very, very deep branches, as you see. So this is what people found in mitochondria. This later has been substantiated by many other ways. Substantiated by Y chromosomes, substantiated by autosomals, substantiated by other kinds of things that we carry like obligate pathogens, like the lice that we have in our hair. If you look at the African lice and the and the non-African lice, they're very different. If you look at the tuberculosis that we carry, the tuberculosis in Africa is very different from the tuberculosis everywhere else. And if you look at the branches, the tree of the tuberculosis, the Mycobacterium, Mycobacterium, the bacterial variation, then that also mimics very similar to how the human tree behaves. So it's very similar to the way in which humans have moved across the different places. So this is something which is called a replacement theory. This is also called something which is a replacement theory. Why is this a replacement theory? Because this is a story of the anatomically modern humans. And when anatomically modern humans moved out, they did not move out in a vacuum. They moved out, and when they moved out, there is very, very strong evidence of coexistence of anatomically modern humans with other kinds of hominids, like the Neanderthals, for instance. So the evidence of the Neanderthals has been as recent as 30,000 years back in somewhere in, suppose, in Europe. And these people definitely moved out of Africa somewhere around 60,000 years ago. So there is a considerable amount of overlap. But as like human history, whenever humans moved or anatomically humans moved, there has been an elimination of multiple species, which included the Neanderthals as well. So this has been something which is called a replacement theory, which is like we as anatomically modern humans, we have replaced the Neanderthals, for instance, in different parts of the globe. In the process, we have possibly also replaced other kinds of hominids who are also living in different places. All right, so these are things. So there are other kinds of evidence from completely different kinds of disciplines, which also came and substantiated this. So the timing calculated from genomic data, which corresponds to the movement of anatomically modern, or the successful movement of anatomically modern humans out of Africa, also corresponds with different kinds of changes in the geology. So different kinds of changes which can be ascribed to the climatic changes which has happened during that time, which means that Africa was going through a huge transformation, and it was kind of habitable for people living here to move to places which are pretty close by, which had a considerably different kind of climate. So the climate change was happening simultaneous to this, which kind of provides an indirect evidence of why people should move out of this. But there are multiple things which was also found from fossil records from a very early time, which is that if you look at this has a lot of these red lines, which is the movement of anatomically modern humans in different parts of the globe, the red lines of that, Homo sapiens sapiens migration. But if you look at the yellow lines were the Homo erectus, who are hominids who lived long way back. And they have been possibly the first people who used fire. They were possibly the first people who might have had a language by themselves, a spoken language of themselves. They were pretty successful, and I'll come to that at some point of time. And there are archaeological evidences, archaeological sites, very, very old archaeological sites, way before the anatomically modern humans even surfaced somewhere. And that archaeological sites actually started in Africa, again went out of Africa. So they went out of Africa. That corresponds to another kind of a geographical epoch which happened at that time, when anatomically modern humans were not there. So it's the same kind of climatic change which possibly has led the movement of the Homo erectus out of Africa, and the same kind of things have also led to the movement of the Homo sapiens sapiens. Now, the timings that people could reconstruct, those kinds of things can actually be ascribed also to different kinds of movements that we have. So, for instance, the movement of anatomically modern humans, the Homo sapiens sapiens to Australia, was pretty rapid. So by 60,000 years ago, from 60,000 years from present, there was definite presence of anatomically modern humans in Australia, whereas Europe was populated only about 40,000 years. So we actually moved out of Africa and reached Australia way before we actually had been successful in colonizing Europe. This part of the world was the one which was populated the most recently, which is only about 15,000 to 30,000. Now, when people moved out of Africa, there were two things. One is, as I mentioned, they did not move out in a vacuum. which means that there were other kinds of hominids who were here, but definite evidence of Neanderthals who were here, and we'll talk about some of the species. It is also a very different kind of a world. So in order to go to Australia, you can actually walk all the way to the tip of Indonesia. And then you still have to cross a little bit of water to get to Australia. But there was a lot of, so this was a glacial maximum, and there was a lot of water which was trapped in these glaciers. So the sea levels were lower. So you could actually walk to different places. You could actually walk across this, but if you walk across this, you immediately encounter a large glacier. And this 30,000 to 50,000 years ago, when people actually moved through this, is essentially also corresponds to the time when this glacier starts melting, which is the timing when things started to stabilize, the kind of the time which is prelude to agricultural revolution or the domestication of human beings, etc. So prior to that, everybody was some kind of a hunter-gatherer. So prior to just 15,000 years ago, we were just moving, migrating, and hunting and gathering. So we were kind of the kind of lifestyles that we are getting adapted to is only a very small proportion of time we are on this earth. It's only about 10,000 years on this world that we are doing this, whereas now the recent estimates of anatomically modern humans evolving in Africa goes around 30,000 years. All right, so the human exodus has been like this, and there has been finer points in which we can come up with timings. One of the earliest places where people moved is India, which is one of the reasons that I find it very exciting to study this part of the subcontinent. There has been evidence of population expansions here, and one of the reasons that in order to go to Australia, you possibly have to travel to some of these places. So this is kind of a link between all of this. Another interesting thing why molecular methods always have advantages over archaeological methods, will continue to have advantages over archaeological methods, is that suppose this was a place where there might have been human settlement. This is under sea. So it's very difficult for an archaeologist to find something from this. Whereas if you look at molecular methods, you can still decipher about history. Not necessarily you can answer the question whether there have been settlements here, but you can decipher about history without even having to look at it. All right, so the question is that all of this is from extant genomes, as we are talking about. Genomes which is like present-day individuals. Genomes which is like species which are present today, right? So the question is, can we do any better? The advantage of archaeologists is essentially that they can really go back deep in time, right? They can go and find a fossil of something which is not existing. That is something which genomics still cannot do, because you do not have the molecular data of those individuals. You cannot have molecular data from the past. So the question that people started, obviously scientists are ambitious beings, and they've started asking is that, is it true that we have to limit ourselves only to DNA that I can collect from extant individuals who are present now? So can I go back to the past? Because DNA is a very stable molecule. So if it is a stable molecule, can I reconstruct DNA of something which existed earlier? So I'll talk about two scientists here. This is the second part of this. Alan Wilson is the professor from Berkeley, whose work we have been talking about. Alan Wilson is the professor from whose lab essentially put forward this out of Africa theory, etc., etc. They are the ones who showed that the humans and the chimpanzees are very close to one another in terms of their genomic sequences. And there is this individual, Savante Papa, who is very famous because he got the Nobel Prize a year or two back in medicine, whereas what he did is essentially studying evolution. His biography or autobiography is known as the Neanderthal Man, and he considers himself, why is he? So I start with Savante Pava's story. Savante was a scientist in laboratories, I think in Sweden, and he was doing his research in immunology. That was his day job. His night job was somehow Savante's mother was an archaeologist, and Savante, so his biological father was a Nobel laureate who never married her mother. Never married his mother. But Savante who used to go to different places with his mother, and his mother was an expert in Egyptology. And Savante somehow, because of those days, Savante had a little bit of mummy in his possession as a kid. So when Savante joined this lab, this biology lab, doing all immunology during his daytime, his nighttime job was trying to extract DNA from the mummies. So if you can do that, then you can actually reconstruct DNA or get DNA from something which lived in the past, which is not like DNA from the present day. So Savante did that, and at the same point of time, obviously, Alan Wilson was also trying to do that. Alan Wilson got this remains from something which is called a quagga, which is a very species which is very close to a zebra, and this was a stuffed quagga in the San Diego Zoo, and Alan Wilson could get a sample from that quagga. And what he tried to do was to reconstruct the DNA sequence of the quagga, which no longer exists, which got extinct. So Alan Wilson's paper was published in Nature in 1984. So Alan Wilson, as you can see, is the last author, senior author in this paper. And in the next year, in 1985, April 1985, this is Savante Pavo's single-authored Nature paper, which came out of the night job that he did. So the day job was immunology. He was pretty successful in that as well, but nothing compared to his night job, which got him a single-authored Nature paper, which essentially reconstructs the DNA of an Egyptian mummy. And once he has done this, he was considering going to Alan Wilson's lab for a postdoc. What else? Naturally, Alan Wilson was an established scientist, so he was considering to go to Alan Wilson's lab for a postdoc, and he receives a letter from Alan Wilson. When he was considering this, he receives a letter from Alan Wilson, and it says that, Professor Pavo, it was a pleasure reading your paper. I think you have done a much better job than myself. If possible, can I spend my summer? I can take a sabbatical this summer. Can I go and spend my summer in your lab? So Savante writes back that that Professor Wilson, this is absolutely impossible. This summer I cannot accommodate you. I cannot accommodate you in my lab because I do not have a lab. In reversal, can I give you a different proposition, which is I go and join your lab as a professor, which happened. It's amazing that Savante and Wilson, they got together, and what they did for the next few years is essentially they proved that what they have done is all wrong. So all of this, in principle, was correct, but this was erroneous. So for the next, so Savante's stint in Alan Wilson's laboratory was about standardization different techniques in which you can do this, getting this from ancient DNA, but it was also in proving themselves wrong. And that possibly is one of the qualities which make both of them wonderful scientists, that they spent the productive years together to prove that they were wrong, absolutely wrong about this. And this is how, this slide I actually stole from Savante. This is what happened. So this is Savante's abstraction of the things. So when they did this, this was like an innovation trigger. There was a huge inflated expectation. And at that point of time, if you look at Nature and Science, then you will find that scientists would come up saying that this is the DNA of an extinct dinosaur from a dinosaur egg. This is a mosquito which lived during that period of time. So all of this, yeah, Jurassic Park possibly is the imagination of this inflated expectations. Then things kind of tapered down. Obviously, the way it works in science is it tapered down to something which is almost like a disillusionment about things. People thought that it will never be able to, people thought that it will not be possible to get DNA out of really ancient things. And then around the time that Rajiv and I, we were trying to do our PhDs at ISI, around that time, there was something else which started happening. So this is about the Neander Valley. As you can imagine, that Neanderthals is is a name derived essentially from Neanderthal Valley, because this is where the first skeletal remains were discovered. Then there are these Vidia caves. Then there are other caves in Europe where there are skeletons which are very similar to the Neanderthals, right? That's how you do this. Now, the caves are fantastic places because the caves are like refrigerators. The caves have very stable kind of temperature. The temperature do not vary. In the last 40,000 years, the temperatures haven't varied too much. And in Europe, these caves are pretty cool caves. They do not have too much of bacteria, so the bacterial contamination is limited. So they have a lot of these relatively intact skeletons. And Savante got a few of these bones from different kinds of Neanderthal remains, and he was trying to experiment with different kinds of things to try and get live molecules from people. So the question that he was asking, there was one kind of question that they were asking, is that really, is there a basis to the multi-regional hypothesis at all, right? Whether it is true that, as some people would propose, that the Neanderthals were like ancestors to today's humans, or what exactly make us humans, right? So what differences that we observe from chimpanzee to us is essentially a difference of 6 million years. If we can get something which lived only around, say, 0.8 million years ago or 1 million years ago, then we can actually get an ancestor who is much, or a cousin who is much closer to that. So can I do that? Can I get to a little bit more closer of what are the steps which has evolved in the process of when we separated from chimpanzees as a species to evolve as an anatomically model? So the questions are that what makes us human, and of course, what have been our relationships with Neanderthals? So this is science breakthrough of year 1997, right? And so this came out in Cell, and these are the authors. As you see, Savante Pabo is one of the authors here. So what they did is that they were convinced now that this is the first time that from those Neanderthal bones, they had to do a lot of things. They had to mix the bones, etc., etc. But they could actually get mitochondrial sequences. They could get mitochondrial sequences of Neanderthals. And Neanderthal DNA sequence and the origin of modern humans is considered as a breakthrough in the year 1997. So what they did was they could find 123 clones from 13 amplifications. Essentially, this is only 379 base pairs. So you think about this, our genomes are 3 billion bases. So they could get a stretch of 379 base pairs from the mitochondria of a Neanderthal. And that particular thing is, as you can understand, that has homologous segments in humans as well. So they compared. They compared the mitochondria of the Neanderthal with the mitochondria of humans. And this is what they got. So very simple. This is the entire data analysis of that paper. So what they did was they compared that. So they had 478 Africans, 510 Europeans, some Asians, and some Native Americans. So these are essentially extant individuals, Homo sapiens sapiens from different parts of the world, and they looked at the differences. So they compared that. So they take a modern human, anatomically modern human, compared that with this 379 faces from these 13 amplifications that they got, right? And they just counted the number of faces that there were differences. And what they encountered is something like this, that modern human sequence among themselves for this 379 region, this region of 379 bases, where on an average about 8 differences were observed. This is the range. If you look at the human and the Neanderthal, that was about 27. So this kind of substantiates the Out of Africa movement, which says that the anatomically modern humans, irrespective of what the world there are about 8 places different. I mean, in this 379, there are only 8 places where you observe the differences. Whereas if you compare that with the Neanderthal, there are about a 3-fold increase in the number of differences. And it also corresponds to something like this. So if you compare the human with the human, then the histogram looks like this, which has a mean somewhere around 8, as you can see. If you compare the Neanderthal and the humans, then this is around 27, as you can see. And if you compare the human and chimpanzee, this is somewhere here. And if this corresponds to 5 to 6 million years, also substantiated by other kinds of records, then this would calibrate to something around 1 million. So this is about 1 million years that anatomically modern humans have separated from the Neanderthals, whereas the modern humans essentially have started moving out of Africa only about 0.3 million years ago. I mean, at that point of time, the general understanding was about 0.2 million years ago. So this is the finding in 1997. Of course, there were many challenges, and they encountered that. But scientists being scientists, and Samantha Pavo, as you can understand, the individual that he is, he was pretty dogged. He was not satisfied at this 379. He was not satisfied at looking at mitochondria, because mitochondria is a very small part of your genome, right? It's a very small part of your inheritance. It's only, so if you just look at 1 generation back, it is half of it. It's just your mother. If you look 2 generations back, it is 1 of your 4 grandparents. If you look 3 generations back, it is 1 of your 8 grandparents. So if you go n generations back, it is just 1 upon 2n, 1 upon 2 raised to the power n, part of your inheritance, essentially. So they were obviously not very satisfied. They tried to do this not only with mitochondria, but with other parts of the genome. And they were trying. And at that point of time, there were other kinds of things. There were technological movement revolutions which were happening in genetics. And one of this was something which is now known as a next generation sequencing. Which is a completely different ball game from the sequencing that used to happen at that point. So if I have a jump cut from that time point, 1997 to 2013, it's around the time that we joined at different institutes as faculties. There was this paper. So remember that from this many clones, they got 379 base pairs. And they got a complete genome sequence of Neanderthals from Altai Mountains. This is 2013. So this is about 3 billion bases of Neanderthal genome sequenced from those things. And by that time, obviously, aided by the technological advancement, they have also modified their laboratory methods, their laboratory techniques. So now they're very proficient in getting data out of ancient DNA. And this means that there has been other kinds of things which are bound to happen, because Neanderthals are not the only remains that you have, not the only ancient remains that you have. So this is so this is another cave in Denisova in Siberia, where this is very popular among these people because they had a lot of Neanderthal remains, and they have fascinating history of how humans have evolved. They have all kinds of animal remains, and etc. So there are different kinds of things which started to evolve. I haven't documented some of this, but people did the DNA sequence of an woolly mammoth. Again, something which got extinct. Then they did DNA sequences of things which live for the back in time. Not only that, as you can imagine, that now you have things to do that you can go to a graveyard, get an ancient skeleton dug out, or an ancient individual dug out from a graveyard, and get the DNA out of it or her and sequence it. Essentially, you can really time scale this, or your time traverse has been made so much more easier if you have this kind of data. So they went to Denisovan cave. This is the name of a cave, and from that cave they found a DNA, which is a small digit from the left this finger of a girl, and that's possibly a Neanderthal remain. So they get the DNA out of the sequence, it, find a completely new sequence, find a completely different species. So just imagine the power of molecular studies, that just by looking at this, just by looking at one single digit of an individual, there is no way by which you can classify what kind of skeleton that is. But if you can extract DNA from it, you can well classify that this individual is very distinct from any other. And of course, not a modern human. This is a completely different species of which there is no other evidence, no other archaeological evidence. And they named this as the Denisovan bird. Then they were doing something in Europe, then they find an individual whose mother is a Neanderthal and a father is a Denisovan. So you have these sequences, and you find an ancient individual, serendipity of course, but you find that individual's mother is a Neanderthal and the father is a Denisovan. So now you can go and ask different kinds of questions. You can ask questions like that these two then do not look like two species which are reproductively isolated. These can have a viable offspring. So if you have this, then you can actually have a viable offspring. The question now you can again raise is that did we have a viable offspring with Neanderthals, for instance? Or the anatomically modern humans, could they have a viable offspring with the Denisovan, for instance? So of course, this is applied to different kinds of other things. This is applied to ancient viruses. This has been applied to deciphering about ancient plague, how the plague was different in the first Black Death compared to the plague which happened more recently. Or if you look at Black Death for that, so you can look at ancient bacteria, you can look at ancient viruses, you can look at things which you could only imagine if you have the technology. Again, so a million-year-old mammoth. This was in Siberia. It was preserved so well they could actually get this DNA out of a million. This is the evidence of the record-shattering oldest ancient DNA. So as we are talking about, so this is the Denisovan cave from Russian Caucasus, and from which the Denisovan cave, as you can see, this is the picture of the cave. You can see these gradings there. So this corresponds to different time points in the past. And from some of these time points, they got this remain, which they sequenced, and they found that the Denisovans are somewhere here. And the Denisovans are actually pretty close to Neanderthals. And if you look at the modern human, if you compare the DNA of all of them, then they look somewhere like this. So the Denisovans were found over them. So it again re-asks the question, what makes us humans, and did we interbred with these individuals? So as we were talking about, that this was what was known about our relationship with our closest cousins. And we now discover cousins which are much closer to that, right? DNA sequences of these cousins, at least. So these are our living cousins, these are our extinct cousins. But we can actually get a sense of where they were, what their genomes were, what kind of diseases they might have been, what did they have language, or were they tall, were they short, were they whatever. You ask the question. So if you look at the human genome, then for instance, now you have references. You have reference of a human genome, anatomically modern human. You have reference sequence of a Neanderthal. You have a reference sequence of a Denisovan. You can just take my genome, for instance, and see whether there are regions of my genome which are more similar to a Neanderthal, for instance, or whether there are regions in my genome which are similar to a Denisovan, for instance. And what you find is absolutely surprising. If you look into African genomes, as a whole, African genomes do not have Neanderthal admixture. There is almost very little evidence of anything which resembles a Neanderthal genome in the Africans. But if you look into anybody else in the world, depending on where they live, there is about 1 to 2% of Neanderthals in all of us. And mind it, this is ingrained in our genomes. This is 1 to 2%. If you imagine this is a small number, but this is going to stay. Because when you pass on your genes to the next generation, you pass this 1 to 2% to the next generation as well. So your next generation will also get this 2% of genome, which is inherited from Neanderthals at some point. So what this tells you is that not only there were instances of mating between humans and Neanderthals, but the more important question is that they actually had viable offsprings. And the viable offsprings could actually reproduce. And that's the reason that this kind of admixture could have actually happened. And if you look into this part, this is where the Denisovans were discovered. But if you look into populations who live in this part of the world, there is an additional about 0.2, 3, 4, 5%, in some places 3%, additional Denisovan admixture in this as well. So you found an individual here who's half Neanderthal, half Denisovan. That's an ancient individual. And you find that there are people here who are living people who have a considerable amount of Denisovan admixture. So the ghost hominid that you discovered, not through a fossil, not through a skeleton, but through a DNA, is essentially existing in our DNAs as well. essentially existing in today's population in a pretty leading form, and that's getting passed on from one generation to another. So the question is that what makes us human, and has we been benefited? They have got extinct, but they are living through us in some ways. But how have that impacted on us? Have they really impacted on us in a sense? So you imagine this from an immunological point of view. So anatomically modern humans, they were living in Africa. There is a climatic change. You move out of Africa, right? You move out of Africa into Europe. So if you move into Europe, there's a completely new set of pathogens, right? A completely new kind of environment in which you are trying to adapt. So your immune system has to adapt to that kind of an environment. And how do you adapt to that? The best way to adapt to that is to steal the immune system or steal components from the immune system of somebody who has done this hard work earlier. So the Neanderthals had done this. The Neanderthals were living in Europe and they were successful in Europe for a very long period of time. So when we moved out, we replaced them, yes, but we also do not replace them just as is. We actually took the HLS from them. We took their immune genes from them, a considerable amount of that. And that gave us advantage in fighting the pathogens of Europe, fighting the pathogens of the out of Africa places where the anatomically modern humans live. So this HLA, which is HLA class 1, which is a vital immune component, our HLA class 1, compared to the rest of our genome, looks much more similar to the Neanderthals. And there is a considerable higher proportion of Neanderthal admixture in the HLA class 1 of people who particularly live in places around Europe. So we have done this. We have replaced people in a sense, but this replacement has been leaky. It has been so that we have replaced, yes, but the genomes have been leaking through one species to another, has been passing across to one species and another. So this HLS story appeared somewhere in, it's only as recent as 2011 it appeared in Science. There is another, so if we talk about the Denisovans, do the Denisovans have a contribution? Denisovans have a very little contribution in our genomes, as you can see. I mean, there are only some populations in Papua New Guinea, etc., where there is about 6% of Denisovan admixture. Everywhere else, it's very close to zero. But this is one of the evidence of places where people have really lived in conditions which are inhabitable to people. So this is high altitude living. There are only 3 places on this world where people continuously live in a very high altitude. This is one of them. This is the best place of this. This is Tibet. And if you look at the Tibetans, they have something, you know, the Sherpas climb the mountains very well compared to anybody else. They have something which is known as a super athlete gene. If you look at the super athlete gene of individuals who live in about 4,000 meters without considerable discomfort, and you can also go and live there for 1 or 2 days, but will have discomfort, then they live in a very hypoxic condition, low oxygen levels. They have this EPAS1 gene, which is the one which is connected with this high altitude living, and this is very, very strong. This EPAS1 gene has a very high sequence similarity with Denisovans. So we possibly took this from Denisovans as well. So I would like to end this with this broad picture of how human evolution has moved. So this is timeline. There has been multiple. So that this has been our common ancestors diverged. One of these group give rise to these 2 groups, the Denisovans and the Neanderthals, as they call them. And then there are the anatomically modern humans. Some of them died off, but they also had a mixture with these people. This is the out of Africa. This is the out of Africa time point where people really moved to different places. And when they were moving to different places, the Denisovans and the Neanderthals were still alive, and they had exchanges. They had exchanges with all of this. They have different proportion of exchanges with all of this. And if you come pretty close to present day, then you see the population expansions where these genes have been ingrained in all of us. So I'll end with, because we started with evolution, I'll end with something which is a very faulty depiction of evolution that we have seen. Evolution really do not work this way. I mean, it's not like a linear process by which people move and they start like this and move to this. It's not like evolution is trying to perfect you as a creature. It's not true. I mean, whatever you make out of this, even this is not true. This depiction of evolution is extremely incomplete and actually misinformative about this. Evolution is more like this, which is a branching process where there has been admixtures across this, and these are trials and errors, some of which are successful, some of which are not successful. And if you look at the human chimpanzees, so the chimpanzees have lived through this separation that we have with them. The chimpanzees have lived through 6 million years. We are a proud species, and we think that everything in the world is designed for us. And we have lived only for this time period, which is very, very small, right? Compared to 6 million years, this is only about 0.3 million years, right? So we have lived through only a very small amount of time. Compare that even with Homo erectus, who had been pretty successful, lived through a long period of time, or many of our ancestors who got extinct, but they lived for a longer period of time. And our existence today is somehow a profound legacy of all things which have happened in between. And there has been some kind of contribution to all of this as we understand it now. So evolution is not a linear process. It has been a process which has profound implications in terms of trials and errors that has gone through. There has been different kinds of admixtures which has happened, and overall people have been benefited by the diversity. So it is also it helps us in understanding science, of course. It helps us in understanding questions about our origins, asking questions about who are we, where do we come from. But I think if you study evolution properly, it also makes you humble in understanding that we have been living for a very small period and we are not very, very significant here. I mean, just a small change, as it has happened previously, a small change might actually wipe us entirely, the entire species, completely from this earth. All right, thank you so much for listening. Okay, it has been 15 minutes over time, but I
Very nice talk.
think you have all been.
Maybe we can have some questions. Yeah. Thank you so much for talking to all the audience today. I was just wondering about the process, because over the years, the technique for the DNA sequencing has been incredibly developed. And now we are in ideal situation with the DNA sequencing. But how was it happening? Like, let's say what papers what we were showing around 1980 and all that. So it doesn't seem to be that easy process by then. And how they were sequencing all that.
Yeah, so this is so around that time. This was like what is called a Sanger sequencing method. So Sanger got 2 Nobel Prizes. One of them was because of the technology that he could develop for sequencing this. So a Sanger sequencing is essentially like this, where you can, at a stretch, develop a sequence something which is around 600 bases. All right. So just to give you a sense of how sequencing technology has moved, so the human genome reference that I was talking about, which is about 3 billion bases, and 600 bases is like a 6-inch scale if you compare the distance between here to Bombay, right? So they are trying to scale something which is a distance from here to Bombay using something which is a 6-inch scale. And of course, there were questions of where you start, because it was not like you knew where you have made this, right? There's no point of marking them. So you have to have considerable amount of overlap and then do this. So the reference sequence, in order to make the reference sequence, people spent somewhere around $3 billion, 3 billion. And it took 13 years. And it took an enormous manpower of people working across the world in 3 different continents, 3 or 4 different continents, actually. And there was an enormous number of laboratories which were engaged to just get one sequence of reference humans in 13 years. So our institute, which is conceived in around 2009, our institute have this next generation sequences. We are not terribly rich. We are a very small institute in a relatively poor, resource poor country. and we have sequenced something which is equivalent to something about 30,000 human individuals. 30,000 in this few years. So this is how the technology has actually moved. So there are comparisons of this with respect to Moore's law, for instance, which is considered as the pinnacle of technological movement or technological development. So if you look at Moore's law, if this is the cost of, or if this is the cost in which it has happened for semiconductors, then if you look at sequencing technology, it is like this. It just goes like this.
Thank you. So this molecular sequencing, is it possible to do for small period of time? It's like there are some theory that where aliens come from.
Yeah.
Can it be answered from this kind of analysis?
You have to call me to give another talk to do that. Yes, I mean, so this is one of the questions that I'm very interested in, and there has been this thing which is, so in Rakigari they got, so there are problems, practical problems here, which is that ancient DNA, getting ancient DNA from places like India or places like Southeast Asia or the tropical regions is very difficult. It's because it is contaminated. There's a huge amount of bacterial action, and all of them have DNA, right? So you have to get the DNA, and you have to be sure that this DNA is from the skeleton, which is a huge challenge. Now, if you have something which is very distinct, then it's relatively easy, and there are ways and means by which people do this. So very recently, there has been this skeleton from Rakigari, which has been sequenced, and the story which come out of the Rakigari sequence, I believe, is very incomplete. And there are lots and lots of nuances about this, but yes, it does, and it can ask questions like this: that were there people who came in? Was that a sudden invasion? Was that a slow wave of migration? And questions.
Sure. I have two questions. One is you mentioned about this genetic key.
Mitochondrial.
Yeah,
yes.
It's not literally one.
Exactly.
Maybe from one community,
No.
the resolution is so low that we can't distinguish. So that's one. The other would be, as you said, Neanderthals had a very less intersection with the African ancestors that we pointed out. So does that mean they themselves had a common ancestor?
They had a common ancestor way back,
but we don't know where they are from.
That's so they had a common ancestor which goes way back.
But geographically, we don't know where they are.
Who? The Neanderthals.
The common ancestor.
Oh, for the Neanderthals and
and say genetically. Genetically and Neanderthal.
No, possibly not. But there is evidence that if you look into the African genome, for instance, African genome also have hominids, but you do not have a reference sequence. So having a reference sequence makes your life easier, right? You can actually look at the sequence and see that this matches. This part of my genome matches with this reference sequence. But there is evidence of, suppose, if you look at the African genome, there are certain regions where it looks considerably different. If you look at a maximum likelihood of this being a human thing, then there is a low likelihood of that happening. And there is something else which is also called recombination, which breaks all of this down. Africans being very more ancient compared to other people, there is recombination which also has an effect. So the chances of discovery in Africa is anywhere low. You do not have a reference sequence, but overall there is evidence that they also had admixture with hominids. So this admixture with not necessarily near some other form of hominids, whom we do.
I have a question that how these morphological changes come in human being, like Africans are different than Caucasians than the Chinese.
So morphology is one of many differences that we have, right? But again, this is so if you look at things, so things which are skin deep or judging by the cover is so superficial. Just imagine things like human skin. All right. So if I look at African and somebody who's from an Andaman Islands, Java, if I look at the skin and look at the expression or the genomic sequence, not the genomic sequence, the expression of different proteins in the skins, what is expected? Morphologically, they are very dark skinned. I'm talking about dark skinned Africans. And Jawas are also very dark skinned. But if you look at the expression of genes, different kinds of genes, then you see that the Jarwa Onge or the Pacific Islanders expression of genes in the skin is closer to what you have in, suppose, the East Asians, the Chinese, but the skins look different. So morphology is essentially a very different kind of a body. It's just really the surface of it, just like scratching of the surface. There are so much more in this, so much more in terms of function, that morphology is just a very minor thing. There are changes which has happened in our morphology. Some of these has been directed changes as well. For instance, the short stature of people who, like what people call are the pygmies, or people who live in different kinds of conditions. People who live in islands, they have short statures, which are definite morphological features. The curly hair, the elevated cheekbones, the color of eye. So many of these has been identified. Many of them has been because of just one or two genes which have changed.
So how long it goes? I mean, suppose they have migrated 30,000 years back.
Who?
The Africans to the Europe, or
Okay. So that Europe has been populated about 40,000.
40,000 years. So when they have populated to the Europe, they were not white.
Why did it exist?
Why didn't they
Possibly,
exist? And
possibly not.
Then how
Possibly
they become, then how they become white?
So this is something which is a slightly different kind of a thing which has happened, some different kind of evolution. This is more like going back to Darwin. So suppose you have a skin which is dark color, right? So if you have a dark color skin, then you do not synthesize vitamin D that efficient. Vitamin D is very directly correlated with reproduction in many ways. So if you move to a place which is cooler, which has less irradiation, then you tend to, so people who have a lighter color skin, they possibly reproduce a little bit more efficiently compared to them as a group. And over time, this essentially becomes a population.
Okay.
And it's pretty interesting that there are populations called Inuits who live in Greenland. They live in Greenland. There are other populations around them. The Inuits have very dark color skin. Everybody else has light color skin. The Inuits actually get their vitamin D directly from the kind of diet they have. So they eat a lot of sea fish, which gives directly vitamin D. So there is no requirement essentially for the Inuits to change their skin. Well, I mean, you do not change it per se, but nature do not see a requirement to change the skin.
Okay. So I think there is no other question. So let's thank Analabha for the wonderful talk. And we can see outside. Thank you.